Hello students and welcome to another lecture in the course Physiological Psychology. Today we'll be looking at the topic synaptic transmission and neurotransmitters. And I thought I'd begin today's lecture by giving you a list of learning objectives. These learning objectives will allow you to focus on what you need to know at the end of this lecture. What you need to know at the end of reading the chapter in your relevant textbook. And it provides a nice checklist that makes sure you have covered the material appropriately. So you're going to see that we start off with some basic definitions and understanding of what exactly synaptic transmission is. From there, you need to be able to describe the anatomy of the synapse. And we will see that that is fairly simple and there's not much to that. And then the rest of the course the rest of the lecture is going to focus on the process of synaptic transmission, how synaptic transmission comes about, which would include how a neurotransmitter is released at the synapse and how they're removed, and what effect that they have on the postsynaptic cell. Then we'll talk about the importance of synaptic integration and give two examples, temporal and spatial summation. And finally, we'll conclude by looking at neurotransmitters and the important role that they play in brain physiology and how neurotransmitter function can be modified by drugs. So let us begin our lecture this afternoon or this morning or whatever time it is you're listening to it. It might be actually 1 a.m. in the morning by just looking or watching this video, which gives a brief overview of the process of synaptic transmission. At the end of this video, we will then take each step inside of the process and explain it in more detail. So just look at this video, watch this video, and try and get an overview and understanding of what is happening during the process of synaptic transmission.
When an action potential reaches the synaptic terminal of the axon, it encounters a region called a synapse where two neurons come close together but do not touch one another. A minuscule gap, called a synaptic cleft, separates the synaptic terminal of the first or presynaptic neuron from the dendrite or cell body of the second or postsynaptic neuron. The arrival of an action potential at the synaptic terminal causes the synaptic terminal to become positively charged and release neurotransmitters into the synaptic cleft. The neurotransmitters diffuse rapidly across the synaptic cleft and bind to receptors in the membrane of the dendrites or cell body of the postsynaptic cell. Each type of receptor in the postsynaptic membrane binds to a specific type of neurotransmitter, and after binding to a neurotransmitter, the receptor causes specific types of ion channels in the postsynaptic membrane to open. As ion channels are opened, ions flow across the cell membrane of the postsynaptic neuron along their concentration gradients. This flow of ions into the postsynaptic neuron causes a postsynaptic potential in the dendrites or cell body. Depending on the channels open and the ions that flow in, synaptic potentials can be either excitatory, making a neuron less negative inside and more likely to fire an action potential, or inhibitory, making it more negative and less likely to fire. An excitatory postsynaptic potential is called an EPSP. An inhibitory postsynaptic potential is called an IPSP. A synapse that produces EPSPs in the postsynaptic cell is called an excitatory synapse, while a synapse producing IPSPs is an inhibitory synapse. Postsynaptic potentials cannot travel far in a neuron. After a few millimeters at most, the ions leak back across the membrane and the signal is lost. However, postsynaptic potentials travel far enough to reach the cell body, where they determine whether or not an action potential will be produced.
So that video just gave you a brief overview of synaptic transmission. So let's delve into it in a little bit more detail now. Well, we can say that synaptic transmission is a process by which nerve cells communicate with each other or with their effector organs. Now, why is this important? Well, previously, in looking at the electrical properties of nerve cells or neurons, we've seen that nerve cells and neurons all have an electrical charge, which is called the resting membrane potential. And we've seen that the resting membrane potential is very important because it allows neurons to generate an action potential. And what the action potential does is it takes a signal from the cell body, more particularly the axon hillock, and it transmits that signal along the axon of the neuron, right to the end of the axon, down to the terminal dendrites. The issue is, when it reaches the terminal dendrites, how does this information get transferred to another neuron or the effector organ, like a muscle or a gland cell? And the process by which this happens is called synaptic transmission. One of the important things to appreciate here is that the action potential must stop at the terminal dendrites because there's actually a gap between a nerve and another nerve cell, or there's a gap between the nerve and the muscle cell. And the body must develop some mechanism to bridge this gap. And this mechanism involves the process of synaptic transmission. And it involves special chemical messengers called neurotransmitters. And so you cannot talk about synaptic transmission without talking about neurotransmitters. So in summary, synaptic transmission is a process by which cells, particularly nerve cells, talk to one another. And it's a vital process. It's the process that allows the brain to function. And as we'll see, it's a process in which human beings have been able to modify by the use of drugs. Now, let's look a little bit at the history of synaptic transmission. The idea that nerves release chemicals and that these chemicals can then modulate the effect of organs is about 100 years old. And the person who was first able to prove this was an Austrian physiologist called Otto Loewi. I'm sure I'm not pronouncing that correctly, but he did a very novel experiment. He took 2 hearts from frogs and he put them in a solution. And then for heart number 1, he stimulated the vagus nerve. And he knew when he stimulated the vagus nerve, it would slow down the heartbeat. He had already discovered that stimulation of the vagus nerve slows down the heartbeat. But his theory was that the stimulation of the vagus nerve was releasing some sort of chemical, and it was a chemical that was then interacting with the heart to slow it down. Now, in order to prove this, he thought that if the chemical was being released by the nerve, it shouldn't go directly to the heart, but it should find its way into the solution. And so he said, well, if I could take some of that solution and put it into another chamber with another heart, but that heart wasn't having its vagus nerve stimulated, because the chemical from chamber number 1 got transferred to chamber number 2, we should see the heart in chamber number 2 also slow down. And he was able to carry out this experiment. And he was able to show very, very clearly that that's exactly what happened. When he took some of the perfusate from chamber number 1, which was generated by stimulation of the vagus nerve, and transferred it to chamber number 2, the heart in chamber number 2 slowed down. And so Otto Loewi was the first person to come up with the idea of neurotransmitters and the fact that nerves release a chemical substance that's important to produce effects on other nerves and on their effector organs, muscles, and glands. So let's look now at what we know about the anatomy of the synapse. Now, the synapse is very, very simple. It represents the area between a nerve and its effector organ, or the nerve and the nerve, another nerve with which it's communicating. In this case, we're looking specifically at 2 nerves talking to one another. The nerve in which the signal arrives is called the presynaptic axon, as can be seen on the image on your right, or the presynaptic neuron. The nerve that is being communicated with, or that's receiving the signal, is called the postsynaptic neuron. Very simple. Presynaptic, before the synapse. Postsynaptic, after the synapse. The space in between the two neurons is called the synaptic cleft. Often we refer to the end of the presynaptic neuron, its terminal where it gets a bit round and bulbous, as the synaptic knob or the terminal button, and you should know that already. Inside of the terminal button are some small circular structures or cylindrical globular structures called synaptic vesicles. And inside of these vesicles, we find the chemical messenger, the neurotransmitter. The neurotransmitter. On the postsynaptic membrane are the receptor proteins. And these are the proteins, or these are the receptors that will receive the neurotransmitter, which will then signal the postsynaptic response. So if you follow my mouse, we can say the synapse consists of the presynaptic neuron, which terminates in the terminal button. It consists of the postsynaptic neuron. It consists of the space in between, which is called the synaptic cleft. In the presynaptic neuron, we have these hollow bead-like structures called synaptic vesicles, which contain the chemical neurotransmitters. And finally, on the postsynaptic membrane, we have receptors, which are really proteins that will then interact with the neurotransmitter to produce a postsynaptic response. This is a very simple structure, and it's one which you should know, and you should be able to draw a simple diagram like this and label it appropriately. Here we see a schematic diagram of exactly what I told you, but it also highlights another important point in the figure on the right, that a lot of energy is utilized during synaptic transmission to especially pump the neurotransmitter into the vesicles. So you often see many mitochondria, the powerhouses of the cells, in the terminal button. And to your left, you actually see an electron micrograph of two synapses. Here you see one with the bulbous end. You can see the small vesicles here. You can see the cleft here, which separates the presynaptic structure from the postsynaptic structure. And it actually looks that there's a second synapse here on the right. So the question is, once the action potential arrives at the terminal button, how does it initiate synaptic transmission? What's actually taking place during synaptic transmission? So let's go through this in a series of steps. And step number one will be the arrival of the action potential at the presynaptic nerve membrane. Now, you will know from previous lectures that the action potential is really a depolarization. It's a reversal of polarity such that the inside of the membrane now becomes positive relative to the outside. And this reversal of polarity, this depolarization, activates a group of channels in the terminal button known as voltage-gated calcium channels. These channels only respond to depolarization. And so when the depolarization occurs, the channels open. And because they are specific for calcium, they allow calcium to diffuse down its chemical gradient and move from the extracellular fluid into the cell. So let's go through that again. First, the action potential arrives. Secondly, the arrival of the depolarization at the terminal button opens voltage-gated calcium channels. Thirdly, calcium moves down its concentration gradient from the extracellular fluid to the intracellular fluid of the terminal button. And the arrival of calcium inside of the terminal button triggers a chemical reaction which causes the vesicles to fuse with the presynaptic membrane and to release their neurotransmitter into the synaptic cleft by a process of exocytosis. Now, this process is actually very complicated, but for your purposes, all you need to know is that the influx of calcium triggers these chemical processes, which causes the vesicles to move from where they're located and fuse with the presynaptic membrane and release their neurotransmitter. And that's demonstrated very clearly here in this diagram. Again, this is our terminal booter, and inside of it you can see the mitochondria and numerous synaptic vesicles. When the action potential arrives, voltage-gated calcium channels open, calcium enters the terminal booter, and it causes a vesicle to fuse with the presynaptic membrane and then release its contents via exocytosis. So it really is quite a simple process. The key events being the arrival of the action potential and depolarization, the opening of the voltage-gated channels, allowing calcium to enter the terminal booter, and then the release of the neurotransmitter. Once the neurotransmitter is in the synaptic cleft, well, by a simple process of diffusion, the neurotransmitter will diffuse across the cleft and bind to the receptors on the postsynaptic membrane. Now, each neurotransmitter has specific receptors with which it binds to. And very often these receptors are part of ligand-gated ion channels. You'll remember I told you that different ion channels have different gates. Some gates open in response to voltage. Some gates are always open and are called leaky channels. And then some gates respond when a chemical messenger or a ligand binds to them. Most of the receptors that we'll be considering are ligand-gated ion channels. And in this case, they can let sodium when, for example, at the neuromuscular junction, the neurotransmitter is acetylcholine. When acetylcholine diffuses across the neuromuscular junction, it binds to a nicotinic receptor, and that receptor lets sodium in and it allows potassium out, resulting in a localized depolarization of the postsynaptic membrane, which, if big enough, can reach the threshold and trigger another action potential in the postsynaptic cell. So we can say that the postsynaptic response can either be inhibitory or excitatory, depending on what receptor the neurotransmitter binds to. This is important because you can't know the effect of the neurotransmitter unless you know what receptor it binds to. Some receptors lead to inhibitory responses, which we would call an inhibitory postsynaptic potential. However, the one that will be most familiar to you is the one I just described, which would take place, say, at a neuromuscular junction, where you get an excitatory response because sodium has entered the cell. And that gives us an EPSP, an excitatory postsynaptic potential. So let's just use this diagram here to illustrate what I just said. Here we have our synapse, we have our presynaptic cell, we have our postsynaptic cell. The action potential arrives, opens voltage-gated calcium channels, and calcium enters the cell. That causes the synaptic vesicles to fuse and release their contents, as illustrated by this pink hue here. And now the neurotransmitter binds to the postsynaptic membrane here. And that allows sodium to enter the cell, which causes a depolarization, an excitatory response, which we call an excitatory postsynaptic potential. However, you can also have inhibitory transmission. Here we have an inhibitory process taking place, as evidenced by the teal or light blue color. And what we get here is a hypopolarizing response, which we call the inhibitory postsynaptic potential. Now, these processes happen very quickly in a couple of milliseconds. And if enough of them happen at the same time, the result is that you can get an action potential being triggered in the postsynaptic cell, or in the case of an IPSP, you inhibit action potentials occurring in the postsynaptic cell. So far we've described the process of synaptic transmission. We've looked at the arrival of the action potential in the presynaptic neuron, the opening of voltage-gated channels. The channels open and allow calcium to enter the cell. The calcium triggers the vesicles to fuse and release their contents. Their contents, the neurotransmitter, diffuse and bind to the postsynaptic membrane. And this binding to the receptors triggers a particular response. The last component of synaptic transmission is the removal of the neurotransmitter. And there are three ways that the body uses to get rid of the neurotransmitter. And removing the neurotransmitter is very important because you need to stop the process of synaptic transmission. You don't just want to initiate communication, you want to be able to stop communication. I guess it'd be like talking to somebody who talks too much. At some point, you hope that they actually stop talking. So at some point, our system needs the neurons to stop talking to one another. Now, there are three ways in which we can do that. The first is the simplest, and it's simply that the neurotransmitter diffuses out of the synaptic cleft, and there's no longer enough to activate the postsynaptic receptors. So simply by diffusion, eventually all the neurotransmitter removes itself from the synaptic cleft. But when we want a more rapid response, the body can do two things. One, it can use enzymes that are either located in the synaptic cleft or in the surrounding cells to break down the neurotransmitter. And a very good example of this, again, works with the neurotransmitter acetylcholine, which is broken down by the enzyme acetylcholinesterase. So as soon as acetylcholine is released into the synaptic cleft, some of it starts to get broken down by the enzyme acetylcholinesterase. And eventually the enzyme removes all the acetylcholine. The byproducts are often taken back up into the nerves to produce more acetylcholine, by the way. So that's enzymatic breakdown. That's another way, the second way in which we can stop the neurotransmitter acting and remove it from the cleft. And the final way is by uptake into the presynaptic terminal or into surrounding glial cells. These glial cells and the presynaptic terminal sometimes have receptors that take the neurotransmitter back into themselves, and in so doing, remove the neurotransmitter from the synaptic cleft. And therefore, that also stops synaptic transmission. So, our three ways of removing the neurotransmitter are: one, diffusion out of the synaptic cleft; two, enzymatic breakdown by specific enzymes; and three, uptake of the actual neurotransmitter back into the presynaptic cell or into the surrounding glial cells. Once that is done, the process of synaptic transmission is complete and things return to normal. Now, students, I want you to be able to describe what synaptic transmission is as a concept, but I also would like you to be able to describe the process of synaptic transmission with an example, just as I've done over the last few minutes with you. So this diagram here simply summarizes everything that we've done. And you can see number one is the arrival of the action potential. Number two is the influx of calcium. Number three is the transmitter release. Number four is the binding to the postsynaptic receptor. Number five is what takes place at the postsynaptic receptor. And number six is actual reuptake process, which allows the neurotransmitter to be repackaged back into the vesicles of the synapse. So this diagram literally summarizes everything that took place. And you know that each one of those processes, those six processes, you can talk about and describe in a little bit more detail. So I want to shift gears now and talk a little bit about synaptic integration. Now, if you look at the diagram on the right, you see what is very typical of central nervous system neurons. Here you see one neuron receiving multiple connections from hundreds of other neurons. And so this one neuron actually has hundreds of synapses associated with it. And this can seem very, very complex. And the brain has to make sense of it all. And the brain does it by a process called synaptic integration. Now remember, these synapses can either be inhibitory or excitatory. And what central neurons in the brains have the capacity to do is they have the capacity to receive all of these inputs and synthesize them to give a coordinated output. And that process by which the inputs are synthesized to give a coordinated output is known as synaptic integration. So let's just look at a couple of examples of how synaptic integration can actually occur. And the two examples we're going to look at are called temporal summation and spatial summation. And rather than reading these definitions on this slide, what I will do, if you want, you can pause and read the definitions yourself. But I'm going to use the image on the following slide to explain what is temporal summation and what is spatial summation. So let's look at the top left-hand corner of this image. Here we see a simple process of synaptic transmission taking place. Here we have the presynaptic neuron, and here we have the postsynaptic neuron. And because of synaptic transmission, when an action potential arrives in the presynaptic neuron, synaptic transmission takes place, and you can record an excitatory postsynaptic potential in the postsynaptic neuron with your electrode right here. Now let's look at this situation in B, where rather than having one presynaptic neuron, we actually have 3 presynaptic neurons. If they all fire at the same time, and these neurons are closely related in space, what happens is there's a summation. Each one of these neurons causes an individual small EPSP. But because they all occur at the same time and they're all related in space, each small EPSP is added to the other to produce a much larger EPSP. And in this way, you might actually cross threshold for this neuron and trigger an action potential. Because this process is dependent upon these presynaptic neurons being related in space, we call this spatial summation. Spatial summation. Now, what's particularly interesting is that these don't only have to be excitatory. Sometimes you can have inhibitory inputs, which would actually make your final response actually smaller. And it's a balance of the excitatory and inhibitory inputs which will determine whether your postsynaptic cell generates an action potential or not. So this example in B is spatial summation. Now let's go down to C. In C, we only have one presynaptic neuron. But in this case, there's a series of action potentials that arrive very quickly, one after the other. Each action potential triggers an EPSP. But before the EPSP is able to return to baseline, the second action potential arrives, and therefore it gets added on top. And before that one can return to baseline, a third one arrives, and that gets added on top even further. And again, in this way, it allows us to reach threshold and potentially trigger an action potential in the postsynaptic cell. This process is dependent on action potentials arriving very close to one another in time. And so this process is called temporal summation. And so these basic building blocks allow central neurons within the brain to carry out synaptic integration. There are some very simple processes of temporal summation and spatial summation. But when they work together on a macro level with hundreds of synaptic inputs, it allows the postsynaptic cell to produce a coordinated response. Okay, let's talk a little bit about neurotransmitters. And I'll begin by showing you a brief video just to highlight the importance of neurotransmitters and to familiarize yourself with neurotransmitters. Remember, we've already identified what neurotransmitters are. They are the small chemical molecules found in the synaptic vesicle, which cause the information to transfer from the presynaptic neuron to the postsynaptic neuron.
We know a little bit now about the communication lines between neurons, that they are powered with electricity. We also know that the messages neurons exchange are chemical molecules trapped inside these holding tanks until an electrical signal frees them. The molecules that move from one neuron to the next are called neurotransmitters. In the last decade, we detected transmitters that carry messages about pain and pleasure, about sadness and joy, about stress and relaxation. No one knows how many of these molecules of mood reside in the brain. There could be hundreds. So far, fewer than 50 have been identified. Serotonin is one of them. It's the brain's workhorse molecule, playing numerous roles, not the least of which is to inhibit our violent tendencies. Noradrenaline plays a Paul Revere kind of role. It sounds the alarm about threats from the outside world and prepares us for fight or flight. Substance P is a molecule that carries messages about pain. In a sense, Substance P is what prompts us to cry, ouch. Conversely, our brains produce painkillers, called endogenous morphines or endorphins. Neurotransmitters play a critical role in normal functioning, and yet it's easy to disrupt our chemical equilibrium. If electrical impulses go awry in the brain, the chemical transactions that are so dependent on them also go haywire. Mood disorders and mental illness can result. Change the level of a single neurotransmitter, and it could upset the chemical balance of the entire brain.
Okay, I hope you do find these videos useful, and they just say many of the same things that I've been saying in a different way, but also the animation and the video is probably very useful for some of you to help capture concepts. Sometimes images can capture concepts in a very concise way that helps us learn. So neurotransmitters are the endogenous chemicals, and by endogenous we mean produced within the body, that transmit the signals across the synapse from one neuron to another. There are many molecules that serve the role of neurotransmitters in the central nervous system, and that's one thing I want you to appreciate. There are several different types of neurotransmitters in the brain, and we actually have discovered neurotransmitters within the last 10 to 15 years. And this slide here just highlights some of the different classes of neurotransmitters. The ones that are perhaps most common and the ones that you hear us talking about are the amine and monoamine neurotransmitters like dopamine, noradrenaline, serotonin, acetylcholine, histamine. But then there are other neurotransmitters that are made up of amino acids. Actually, the most abundant neurotransmitters in the brain are the excitatory amino acid glutamate and inhibitory amino acid GABA, which stands for gamma-aminobutyric acid. So glutamate, aspartate, GABA, and glycine are all examples of amino acid neurotransmitters. And amino acids, as you know, form peptides or proteins. And so we have some peptide neurotransmitters like opioids, the endogenous opioids like endorphins and enkephalins, and also oxytocin. And finally, like I said, within the last 10 to 15 years, we've found some novel classes of neurotransmitters made up of very small molecules like nitric oxide and carbon monoxide, and more recently, what are known as the endocannabinoid neurotransmitters. These are marijuana-like substances or THC-like substances that actually exist in the brain, and we'll discuss these more in another setting. So I just want you to appreciate that we have many different neurotransmitters in the brain, and these can be grouped into different classes. Now, there are a number of criteria that allow a substance to be called a neurotransmitter, and they're just listed on these pages for you. These are very self-explanatory. Number one, it must exist in the presynaptic terminal, and that just makes total sense. If it doesn't exist there, then how can it be a neurotransmitter? Number two, the enzyme for making it must be within the presynaptic terminal. Number three, it must be released when a nerve arrives at the presynaptic terminal. Number four, there must be receptors for it to bind with on the postsynaptic membrane. Again, these are very self-explanatory, but they allow us to determine whether a new substance actually functions as a neurotransmitter. And the last two criteria is that when we experiment with these substances, we can replicate the response at the postsynaptic membrane, or we can block the response by giving certain drugs. So scientists who study brain physiology and brain pharmacology are interested in knowing what exactly is a neurotransmitter, and they've come up with these criteria, which are really very, very simple and self-evident. Now, one of the things you need to appreciate about neurotransmitters, and we've said it already, is that neurotransmitters bind to receptors. But in many cases, neurotransmitters have their own populations of receptors, and they actually bind to more than one receptor. So, for example, one of the most common neurotransmitters that you hear us talk about is acetylcholine. And acetylcholine is the neurotransmitter that's released at the neuromuscular junction. That's the junction between the nerve and your skeletal muscles. But interestingly, the receptor found at the neuromuscular junction is the nicotinic receptor. But in other parts of the brain, there's also acetylcholine being released, but it binds to other types of receptors and other parts of the body, which are called muscarinic receptors. And so acetylcholine actually has two types of receptors, or two subpopulations of receptors, nicotinic receptors and muscarinic receptors. And if you were actually to study this in more detail, you'll discover that there are even subtypes of nicotinic and muscarinic receptors. Almost every single neurotransmitter has populations of receptors. So adrenaline has alpha and beta receptors. Dopamine has D1, D2, D3, D4, D5 receptors. And what this multiplicity of receptors does is it allows one neurotransmitter to have multiple effects depending upon which receptor it binds to and what part of the brain it works in. And this, you will appreciate, then makes CNS pharmacology and understanding what's happening in synaptic transmission much, much more difficult, because things are not quite as simple as they might actually appear. Now, one of the things you should start to appreciate now is that because neurotransmitters are chemicals, we can mimic the actions of neurotransmitters or block the actions of neurotransmitters by inserting chemicals into the body. And the chemicals that we insert into the body to do this will be known as drugs. Now, when a drug binds to a receptor and has the same response as the neurotransmitter, we call it an agonist. It's mimicking the effect of the neurotransmitter. But sometimes we want to block the effect of a neurotransmitter and we give another drug. In this case, when it blocks the receptor, we call it an antagonist. And so you can talk about drugs that occupy receptors and activate them versus drugs that occupy receptors and block them. The ones that activate them mimic the action of the neurotransmitters. The ones that block them inhibit the action of the neurotransmitters. And then sometimes you can even have drugs that actually have a mixture of both types of properties, but we won't get into those things. Okay. So I want you to appreciate the terms agonist and antagonist and understand what they mean when you see them. Now, this diagram here is not meant to confuse you, but it's just meant to show you that in the process of synaptic transmission, drugs can act at many, many different places. And so the whole pharmaceutical industry is focusing on how can we find different drugs to act in different places to help modify the functioning of neurons. And I just wanted you to look at this diagram and recognize the amount of research and interest that is taking place in the world today to discovering drugs that help regulate synaptic transmission inside of the body. And we can see some basic examples of why this is important. Let's consider BOTOX. Now, I'm sure many of you have heard of BOTOX because BOTOX has become very popular in the last 10 years for removing wrinkles. But that really depends upon the properties of a particular toxin that blocks the acetylcholine neurotransmitter. So let's just look at this little video that describes that in a little bit more detail.
A friend of mine just got Botox injections for her migraines. Doesn't that sound kind of bizarre to you?
Botox is a little bizarre, but it has great therapeutic and cosmetic benefits. When I first read about this potent nerve toxin in microbiology class many years ago, I never dreamed one day I'd be injecting it into my patients. Produced by the bacteria Clostridium botulinum, it can cause paralysis, respiratory arrest, and even death when swallowed. But it's usually safe when injected into a muscle. It works by blocking the release of acetylcholine, a substance necessary for muscle contractions. In 1989, the FDA licensed Botox for the treatment of different muscular movement disorders. Since then, it has been studied and used for lots of other medical problems. So, in addition to preventing migraines, it reduces severe muscle spasms, stops excessive sweating, tones down tremors caused by Parkinson's disease, and much more. People who have had whiplash get injections to improve the range of motion in their necks. Sufferers of restless leg syndrome often rely on Botox for a good night's sleep. I could go on and on with more examples of how botulinum toxin is being used, but the bottom line is that the drug is not just for erasing wrinkles. So, if you have a condition that you think might benefit from botulinum toxin injections, talk to your doctor. From Harvard, I wish you good health.
So I hope that video was useful, and I hope you were able to appreciate that this compound called Botox, which is adapted from a naturally occurring toxin, has many uses. But the only reason we're able to utilize this compound, which can actually kill you if not properly utilized, is because we understand the process of synaptic transmission, and we've been able to modify the process of synaptic transmission. And that's the goal of many pharmacologists, and the goal that many researchers, as they seek to deal with many debilitating diseases like Parkinson's disease and Alzheimer's disease and schizophrenia. Many of these conditions are the results of problems with neurotransmission, and scientists are actually actively searching to resolve these by coming up with new and different drugs by understanding the processes of synaptic transmission. Here are a couple more examples. Tetanus, a terrible disease. You've probably been vaccinated against it. The tetanus toxoid impairs synaptic transmission. And then the poison from the black widow spider also interferes with synaptic transmission. And I'm just showing you some simple examples from nature to show you how vital synaptic transmission is and how when something goes wrong, it can have catastrophic effects upon the body. And this should just trigger in you maybe a desire to read a little bit more about synaptic transmission and the different drugs that can modify synaptic transmission going forward. So that ends our lecture on synaptic transmission and neurotransmitters. We're now really getting into the meat of physiological psychology and how understanding the physiology and the behavior of neurons, we can now use this to build up to actually understand how do these things influence behavior. And that's going to be one of the really interesting parts of this course as we begin to see how these neurotransmitters that we spoke about in today's lecture actually can change behavior going forward. So until next time, goodbye.